Good day, Year 9s, and welcome to our next lesson on osmosis. Now, last lesson we focused on what osmosis was, looking at the different types of solution and what happens to animal cells. In this lesson, we're going to be more focused on what happens to plant cells, and we're going to be starting off a required practical, thinking about a required practical to do with osmosis in plants. Now, as I said in the last lesson, in this one there is an accompanying video by Miss Corbett which goes over the required practical for osmosis. So please can you complete this lesson first and then move on to that video to show what happened in Miss Corbett's required practical. Okay, so what do we need to know in this lesson? So because we're focusing on plants, we need to be able to understand the importance of osmosis in plant cells. So what happens to plant cells and why that is important. Quite a lot of the success criteria relates to the required practical. So we need to make sure that we're able to know how to plot a suitable graph. We need to be able to look on that graph to pick out where isotonic solution is, so the same concentration inside and outside the cell, and then we need to look at why that's important in plants. Just a quick recap from last lesson. We should have this in our notes, but just a refresher. Osmosis is the net movement of water molecules going from an area of high water concentration to a low water concentration. And that happens through a partially permeable membrane. Now, remember, osmosis is a specific type of diffusion, so it is only happening with water particles, and it has to happen through a partially permeable membrane. Both processes are passive, so neither requires energy in order to move those particles from one place to another. If you can't remember what a partially permeable membrane is the membrane that has small holes in it which allows some molecules, i.e. small molecules, to pass through it, but not other molecules like large molecules, which you would have seen in the practical that Miss Corbett videoed using the Visking tubing, because starch is unable to leave the Visking tubing, but iodine is small enough to go in, which coloured the starch black. Okay, so osmosis in plants. When we looked at the osmosis in animal cells, we saw that because animal cells don't have that cell wall, it means that if too much water goes into the animal cell, it causes it to burst or lysis. And if too much water is leaving the animal cell, then it causes it to shrivel in a process called crenation, because it's unable to fulfill its role anymore. When water moves into a plant cell, the pressure inside it increases. So because it has got this cell wall going around the outside, it means that it doesn't burst, so the pressure inside increases. And we call that pressure the Turgor pressure. So the more pressure, we say, the higher the Turgor pressure. And this pressure helps support the cell and keep them rigid. Now that's really important in plant cells because plants don't have a skeleton like we do. So the way that they support themselves is by having these rigid cells stacked one on top of the other. They've got that regular structure, so it's kind of like a brick wall with all the bricks being the cells that keeps the plant upright. As you may see in summer, if a plant doesn't have enough water, it causes it to wilt, and that's because there's not enough Turgor pressure in those cells. The cell is strong enough to support the pressure and prevents the cells from bursting. And if a cell has got this pressure, this Turgor pressure inside it, we say that the cell is turgid. Okay, so a turgid cell is where the water has moved into the cell and it has caused it to become rigid. So Turgor pressure, rigid, turgid. Now, the opposite of that, when the water leaves the cell, as we said in the last lesson, it causes that vacuole to move away from the cell membrane. So the opposite to turgid is flaccid. So flaccid is where the water is leaving the cell. The cytoplasm moves away from the cell wall, and that's called plasmolysis. And that causes the plant to wilt. So when there's not enough water in the plant, such as in summer where it's very dry, it causes plants to wilt because they don't have that turgor pressure, and the cells become flaccid because the water is leaving the cells. So here we've got two pictures, one from the microscope. One, the top right-hand side one, is showing cells that are in a solution with high water concentration, so a low solute concentration. And what do we call that? Hypertonic solution, that's right. If you look at the one on the left-hand side, these are cells that have been pictured where they've been put in a high concentrated solution or hypertonic solution. So that has caused the water to leave it. And you can see that cell, that cytoplasm moving away from the cell. So what I'd like you to do now is kind of put everything that we did in last lesson along with this lesson, and produce a mind map or a fact sheet or just a comparison, so split your page in half, however you want to do it, to show osmosis in plant and animal cells. So what happens in the three different types of solution: a hypertonic solution, a hypotonic solution, and an isotonic solution. I'm going to put on the next slide a success criteria. So work through that to make sure that you've included all of the things that you need to include in your piece of work. You might want to go and do some further research, so there is a link underneath to the BBC Bite-Size website that just has a little bit of revision on diffusion and osmosis, so you can put it all together. So here is the success criteria. So the first thing that we need to make sure that we have is a labeled diagram showing animal cells in isotonics. That's where the concentration is the same, the solute concentration is the same both inside the cell and outside the cell. In a hypertonic solution, so that is where the outside of the cell has a much higher water particle concentration. Sorry, I've just mixed that up in my head. So in the hypertonic solution, if it's in a hypertonic solution, that means it's got a high solute, so a low water concentration. Took a little while to process that. Hypotonic solution is when the cells are in a low solute, therefore a high water concentration. So I do understand how annoying it is to get it right in your head, because sometimes I can't even get it straight in my head. The next thing you need to do is the same, but this time with plant cells. So you're looking at what happens when water leaves the cells, what happens when water goes into the cells. Can we make sure that we're including those key words about Turgor pressure, when the cell is turgid or flaccid or plasmolized? If you can try and get as many of those words in, that would be brilliant. In securing, you need to be able to explain the changes that take place in animal cells in isotonic, hypertonic, and hypotonic solutions. So we need to be talking about what happens to the cell when too much water enters, too much water leaves. Same again for plant cells. And then finally, to top it all off, evaluate. So that's where you give the advantages and disadvantages and a conclusion. Evaluate why plant and animal cells behave differently in terms of osmosis. So you can link that to why it's so important, this process in plants, keeping that high turgor pressure. So if you could pause the video, have a look on that link so you can do a little bit of revision, add that to your work. But I want a comparison between the animal cells and the plant cells. I will speak to you in a bit. Okay, so we're going to move on to the required practical now. And this is a very commonly asked practical to do with osmosis in plant cells. In a previous exam, it caused a lot of confusion because all the practicals that tend to be done in schools are to do with potatoes, but the question in the exam was about carrots, and a lot of students weren't able to apply the knowledge of what they did with potatoes with carrots. So do bear in mind that this would happen with all plant cells. So the aim of the investigation is to look at what happens with osmosis in potato. So the potato is what Miss Corbett will be using in her video. Variables, what things are going to change in the investigation. So do remember, we have our three variables, the groups of variables. We have the independent variable, that's what we change. We have got dependent variable, that's what we measure. And control variables are things that we have to keep the same. And the more control variables, more things that we are controlling, the more valid our investigation is. So these are some different things that we could look at with osmosis. Now, temperature, because it links in with diffusion, temperature would affect the rate of osmosis. The higher the temperature, the faster those particles are moving, so the quicker it would be. The amount of water, now I hate the word amount, please never use the word amount in your exam answers. We should be putting concentration, volume, etc. But the amount of water, which links in with the one at the bottom, the concentration of a sugar solution, would change the concentration. The size or the shape of the potato chip, so that's what we're going to be looking at, how if the water is moving into the cells, then it's going to affect the size or the mass of the potato chip, and vice versa, if the water leaves, that's also going to affect it. And the pH. So normally, before an investigation, we should have a hypothesis, or what you might have done in previous years as a prediction. So the hypothesis is where you take the independent variable, whatever it is that you are changing. In this case, it's going to be the concentration of sugar solution. And you have to link it, relate it to the dependent variable, what it is that we're measuring. So the mass change of the potato. So you could say that as the sugar solution gets stronger, or as the sugar solution increases, would be a better word, more or less water will move into or out of the potato, and that will cause the potato to lose or gain mass. So can you have a go at putting down a hypothesis for this practical, prediction for this practical, saying, completing the sentence, that as the sugar solution increases, and then pick the words to write that hypothesis. Okay, so pause the video and write that hypothesis. Okay, so hopefully you have written your hypothesis of what you think is going to happen. So you might have said that if you increase the sugar solution, then more water will move into the potato and cause it to gain mass. Or you might have said that if you increase the solution, less water will move in, and therefore we will lose mass. It doesn't matter on the hypothesis, as long as we have thought about it and thought about why that would be. So you might want to explain your prediction and say why you think that that is going to happen. So the next stage that we move into when doing an investigation is testing our prediction, so testing our hypothesis. And we do this by coming up with a method. So a method is a way in which we can test the hypothesis. Normally it will be obviously a practical investigation. So you can have a go at thinking about what apparatus or equipment you would need for your investigation. So we're looking at how different sugar solutions will affect the mass of a potato chip. So what are we going to do, what are we going to need rather, to investigate that? Have a go at drawing a labelled scientific diagram. So it's not a piece of artwork, it's single line drawings to draw how you would set up your equipment. Then we do a step-by-step set of instructions. So this is what you'll be used to in a practical, where you have your method and just step-by-step instructions of what you need to do. How will you increase the reliability of your investigation, or increase the reliability of your results? Well, normally we would do repeats, so we should do things at least 3 times, and from that we would calculate our average, such as a mean. What are we going to do to increase the accuracy? Make sure the measurements are true. So an accurate investigation gets results that are close to the true value. So you can have a think about what we can do to do that. So if you could pause this video, and if you could jot down some ideas for those 5 different things, and then we'll start having a look at the practical. Okay, so hopefully we've had a go at thinking of our own method, and it is important that you're able to do this, because in the exams you may be asked to come up with your own method, or suggest equipment to use, or draw a diagram, and you'll definitely be asked about repeatability and accuracy. So do make sure that you are practicing these as we go. Now you've got 2 choices here. You can either exit this PowerPoint and go and have a look at the practical at play with Miss Corbett's video, or you can continue on here where I've got a question, a 6-mark question, which you can have a go at answering, that's looking at the importance of osmosis, and then do the video afterwards. It's entirely up to you. Okay, so you've either gone and had a go at the practical. I hope you enjoyed that practical and had a look at how you could increase the validity, the reliability, etc. of the practical. What we're going to do now is just a 6-mark question that suggest and explain why osmosis is so important in the structural support system of plants. Now you'll notice that there are 2 command words here, suggest and explain. If you see suggest in the exam, it means the examiner is not expecting you to know an answer, but you just have to give a logical opinion or a logical answer of what you think it will be. And if whenever we're explaining something, as you know, we need to say why it's happening. So you're probably going to have a because in your answer at least once. So I'm going to ask you to pause this video now and have a go at that 6-mark question, suggest and explain why osmosis is so important in the structural support system of plants. Good luck. Okay, so we're going to have a look at your answers, or you're going to be having a look at your answers to see how you have gotten on. You don't have to use a different coloured pen. You have one, brilliant, just makes it very clear what you still need to work on. So from the question, suggest and explain why osmosis is so important in the structural support system of plants, you can see that underneath there is an ideal answer and where you're going to get different marks for it. So plants rely on osmosis to support stems and leaves. So if you've said anything in your answer about it supporting or keeping upright the plant stems, or keeping upright the leaves, keeping open the leaves, then that would be a point. If you said about water moving into the plant cells by osmosis, then that too would be a point. The vacuole swells and presses the cytoplasm against the plant cell walls. So you might have said that when water enters the plant cells, it increases the turgor pressure, or increases the pressure inside the plant cell, making it rigid. The next mark, you would be talking about turgor pressure again. So turgor pressure is reached when pressure is so great that no more water can enter the cell. So when no more water can enter and it has maximum amount of pressure, then that would be another mark. If you said about how that makes the cell rigid or turgid, then that will be a point. And then anything about preventing wilting would be your last point. So hopefully you were able to pick up a few points on that one. Don't panic if you didn't get all 6 marks, but make a note of what it was that you were missing from your answer. Now this brings us to the end of osmosis in plants and the end of osmosis in total. So if you have not yet seen the video by Miss Corbett on the osmosis required practical, please can you make that the next place that you go to, because that is a very important one. If you have already seen it, then I will see you next time, where we're going to be talking about the final way in which substances move in and out of cells, which is active transport. I hope you're all well, and I will talk to you soon. Thank you very much.